Okay, this is a really good question here. So, a research team is analyzing a structural and functional properties of different types of bacterial RNA. After lysing staph A, they see inside of an RNA molecule that is like 90 nucleotides long and has a high saturation of modified bases. And it's in this weird clover leaf shape. So this should be like a little dog whistle that we're dealing with tRNA, not mRNA. And that's going to guide our answer choice here. So this is a start codon. I think it's for like methionine for an mRNA, but we're looking for a tRNA. And we'll explain what that is in a second. But a tRNA molecule is always going to have these acceptor codons of CCA. Okay, so the secondary structure of a tRNA has an amino binding site on OH, and then an acceptor stem, and then a T loop. There's this. So here's this like acceptor stem, and then there's a T loop, and then we go down and we see like the anticodon loop, and then the D arm. And so this is what tRNA looks like. So what's going on here? Yeah, we got some weird modified letters, and it folds into a special shape like a clover. So it tells us tRNA. And that's like a delivery truck for amino acids. So proteins are built from amino acids, and it's the tRNA's job to bring them to the ribosome. So it delivers them to the ribosome where they can be made. And tRNA has a 3' end, which always has the letters C, C, A. Every single time you see tRNA, it's going to start with the CCA here, okay? And so, scrolling down here, that's like the hook on a crane. Without it, you can't pick up the cargo, which is the amino acids. Kind of a cool little analogy there. So that right there, the CCA is like the hook on the crane. Okay, so this tRNA has to have the CCA, the 3' end, so they can grab it. So it's going to be true of bacteria, humans, and every living thing that exists, really. And so that's the right answer. The other answers, AUG, yeah, this is coding for, I believe, methionine. It's the start building thing for mRNA, messenger RNA. And that's what actually tells the ribosomes, hey guys, let's get this thing underway. PolyA tail, that's a safety shell for the mRNA, and it's not found in tRNA. So they add a bunch of A's at the end in order to stop it from breaking down way too fast. So that would be a messenger RNA thing, the PolyA tail. The 5' methylguanosine cap, that's like the passport from the mRNA, it's not tRNA. They add a chemical tag to the mRNA to make sure that it can leave the nucleus and find a ribosome. Okay, so that's the 5' cap, and then the 3 PolyA tail. So yeah, bacteria don't even have a nucleus, and so the tRNA does not need this tag, importantly, because this is bacteria we're dealing with. Ta-ta box, this is part of the DNA, not mRNA, where you say, hey guys, let's start making the RNA here. It's the ta-ta box. It's like this weird little TATA little box here, and what it's going to do is it's going to say, this is the place that you start. Let's make this RNA here. So it's part of the DNA, not even mRNA. UAG, you are gone. This is one of the stop codons. You are annoying. You are gone, for example. These are stop codons in mRNA that say, hey guys, the protein's done. Let's stop this. So tRNA helps the amino acids build the proteins by taking them to the ribosome. It always ends in CCA. Let's go up here. Right up here. It always ends in the CCA or the ACC from the 3' end. Always ends every single time, because that's the acceptor part, so that you know how to pick up those amino acids. And so that's the correct answer. And so we kind of gone over these, but another thing is 90 nucleotides long, and the typical tRNA is 74 to 93. tRNA has these modified bases here, and the cloverleaf structure, all that kind of cues you into that answer. And yeah, these are features of mRNA. So hopefully this was helpful. A good little kind of primer into what tRNA is and what mRNA is, because that stuff is confusing. Like and subscribe if this is helpful.